Okay, it's my pleasure to introduce the third speaker today. Lauren Appleby is an intern, has been a research intern in the Predator Prey program since October 2024. Lauren is from Asheville, North Carolina. She comes to us with a degree, a 2022 degree in environmental science from University of North Carolina, Wilmington. After which she did a stint at Loma Barbadal, tracking monkeys for hours through the tropical forest there in Costa Rica. By all accounts, a grueling experience. After Loma Barbadal, Lauren went on to do her master's degree at University College London, where she looked at the let me get this right. Looked at the influence of landscape heterogeneity on the risk potential for zoonotic invasion, zoonotic disease. And so yeah, that was immediately before coming back to Asheville, right the day after Hurricane Helene last fall. So Lauren has really overcome some things to get to us. While she's been here, she's been she's gone above and beyond as an intern, contributing to a bunch of different projects. So this past spring, we were very proud to have Lauren present a poster at the TWS Florida chapter meeting in Tallahassee, where she analyzed the use of camera traps as a proxy for counting cows in pastures. Kind of an interesting project there. She's contributed a lot to studies of looking at biodiversity and species richness indices using our camera trap data set from Buck Island Ranch. And today she's going to talk about her independent project, Bats of Suburbia, looking at bat community composition across an urban gradient. After her internship, Lauren is in short order off to begin her PhD studies at at Queen's University of Belfast, where she's gonna begin work on the zoonotic potential of African mole rats. So some very interesting work coming up. Lauren's got a theme in her career where she's very interested in zoonotic disease, and so the focus on bats for this intern project seemed like a decent one for us to go with. We're very excited to see what she has for us today. Lauren.
Hi, everybody. As Joe said, I'm Lauren, and I'm really excited to share my intern project with you all, which is called Bats of Suburbia, exploring bat community composition along an urbanization gradient. So Florida is developing really quickly. It's the second fastest growing state by population in the United States. About 1,500 people move here to Florida every single day. And that means that there's increased conversion of natural areas into neighborhoods and businesses and roads that can support this growing population. And a lot of the work that we do in the Predator Prey Lab looks at how development is influencing different species. And so I was really interested in expanding on the scope of this work and looking at a taxa that hasn't really been studied much here at Archbold or in the Predator Prey Lab before, and that is bats. So bats are of the order Chiroptera. They're the second most speciose group of mammals, coming second after rodents, which means that there are a lot of different bat species. They are keystone species due to their role in insect predation and pollination. All the bats that I'm going to be talking about today in Florida are insectivores, so they eat insects and they use echolocation to find their prey and to avoid structures while they're flying. So I was really interested in studying bats, but as I said, there hasn't really been much bat work here at Archbold before, and so I reached out to a collaborator called Dr. Rhoda Petrick. She works at UNC Chapel Hill and is director of Highlands Biological Station. She's done work on urbanization influencing bat activity and also forest edge effects on bat activity. So I was really excited to get the opportunity to collaborate with her on this project. So what bat species should we expect to see in our area? There's two families. There's Vespertilionidae or Vesper bats and Melocidae or free-tailed bats. One morphological difference between these two families is that the free-tailed bats have tails that protrude from their tail membranes, which is how they get their name. I'm interested in all the bats on this page, but particularly these two, the tricolored bat and the Florida bonneted bat, and that's because they're endangered. The tricolored bat is actually state listed, so it's not federally endangered, but it is up for listing. And that is due to white nose syndrome, which is a fungal disease that disrupts hibernation and causes starvation, which is causing really sharp declines in tricolored bat populations. Florida bonneted bat, it's estimated that there are fewer than 1,000 individuals remaining. When you compare that to the Brazilian free-tailed bat, which is the most abundant bat on this page, there are bat houses at the University of Florida, and one bat house can house up to 500,000 Brazilian free-tailed bats. So compared to 1,000 bonneted bats, that's quite a big difference. So again, interested in all of these, but I just wanted to highlight those two in particular. So what were my research questions? One, I want to know, how does urbanization affect bat activity? Are bats influenced more by environmental factors or urbanization and human presence? And how is bat community composition changing along an urbanization gradient? And I did that through an acoustic monitoring study. So these are bat monitors. As I mentioned, bats echolocate to find their prey. And so these bat monitors have built-in microphones that can detect when bats emit these frequencies. And then we can use that data and they'll tell us what bats were found at a site. So moving into the methods, this was the study area I chose. I chose Highlands County, or Lake Wales Ridge on Highlands County, and I overlaid this cooperative land cover map by FWC to get the habitat types in this study area. It's hard to see that, so I zoomed it in a little bit. For those of us who are local to the area, you might recognize Sebring. This is Lake Jackson, US 27. And then I put this 5-kilometer scale on here because bats can travel quite far to forage, so 5 kilometers or even more in a night. So they might not particularly care about these really teeny tiny habitat types on this map, but more the overall area, kind of their surrounding environment. So I wanted to classify these 230 habitat types into urban areas, low modified areas, open water, and then closed water. So I went through all of them very meticulously, and this is sort of what that looked like. Up here, these are open water areas, closed water areas like basin marshes and swamps that are covered by canopy, and then urban areas. So I classified that as anything that had impervious surfaces. So this little snapshot shows commercial and services, communication, and community rec facilities. So after I went through all of those, the map looks like this. So we have low modified, closed water, open water, and urban areas. After that, I generated 30 completely random points on this map, and again, zoomed in just a little bit so you can see 5 of them. I placed a 500-meter buffer around each point to give me a little bit of flexibility on where I could place the monitor, and then I put a 5-kilometer buffer point, or buffer circle, around each point so I could extract the percentage of urban area within each circle. And then I ordered them from lowest percentage to highest percentage and bucketed them in groups of 10. So 10 for each of these categories, buckets 1, 2, and 3. And this is kind of what that looks like. So on the left, you can see this would be our least urban site. And this was a basin or a bayhead that we had to hike into to place this monitor. So it's very far from any kind of development or human presence. In this second bucket category, this was a dirt road in a neighborhood in Sebring. So there's a little bit of tree cover, but it's in a relatively quiet neighborhood. So that's that middle of the road category. And then this third bucket is the most urbanized category. It has the most percentage of urban cover. And you can see it's facing an orange grove, but behind us, there's quite a busy urban center, and this is on a paved road, and a lot of cars drove by. So yeah, that's a little bit what that looks like. Again, there's 10 sites in each of these categories, and we sampled 3 at a time for 10 nights. 10 nights is recommended to detect those endangered species that I was talking about before. So after that was done, I did acoustic data processing, where I put in all of the audio files into Kaleidoscope Pro, which is a software that automatically tells us which audio corresponds to which bat. And so now we can answer these questions. So how does urbanization affect bat activity? Well, as you can see in our most urbanized bucket, bucket 3, actually it had the most bat passes, and our least urbanized bucket, or bucket 1, had the fewest amount of bat passes. Next, are bats influenced more by environmental factors or urbanization? I chose 3 environmental factors: distance to water, tree cover, and temperature. And I did negative binomial generalized models and found no significant relationships between any of these environmental factors in the predicted number of bat passes. And what I mean by a bat pass is one of those calls, the monitor would count that as one bat pass. But if we look at urbanization, so again, this is a percentage of urban cover at each site, I saw a significant increase in the predicted number of bat passes. So as you increase your urban cover, you're increasing the number of, or the bat activity at a site. And my last question, how is bat community composition changing along an urbanization gradient? This is an NMDS plot, or non-metric dimensional scaling. And what that means is these axes are they're arbitrary. We're looking at the proximity between the points. So points that are closer together means that the community composition of those points is similar. What I mean by community composition is the species richness, so the number of different species we detected at a site, and the number of bat passes of each of those species. So if we were to see the green points clustered together and the orange points clustered together, then that would suggest that the bat communities in urban spaces are more similar. But we're not seeing that at all, really. We're seeing a very widespread, there's no pattern. And so that suggests that urbanization is influencing bat species in really variable ways. So taking just a quick look at that, this is the total bat passes per species by urbanization bucket. But I'm going to remove the Brazilian free-tailed bat because they're really abundant, so it kind of muddles it. So just taking that away, you can see the other ones a little bit better. So as you can see, all of these bat species increase their number of bat passes as you go up the urbanization gradient, but some, like the Ori bat and the northern yellow bat and the tricolored bat, increase a lot, whereas others don't increase as much. And so that's kind of what I mean by these species being affected by urbanization in different ways and at different magnitudes. So to wrap it all up, we did all this sampling. What does it all mean? So there's a strong positive correlation between urbanization and bat activity that I found in this study. There's inconsistent differences between bat community compositions along the urbanization gradient, and it suggests that urbanization is affecting bat species in highly variable ways. So going forward, something that I would really like to do is look at the bonneted bat in particular a bit more closely. These calls have to be manually vetted by a trained professional, and I just didn't have enough time to manually vet 1,600 bonneted bat calls before this presentation. So I really want to do that moving forward and look at the trends of that bat in particular, as I mentioned, endangered, and then continue this work in a manuscript for publication and continue my collaboration with Rada. So with that, I would like to thank everybody who helped make this project possible. I'd like to give a special shout out to all of the landowners and business owners that allowed me to place a bat monitor on their land or in their backyard. And yeah, I really enjoyed my time here at Archbold. Thanks so much for listening. Yes.
Well, what season of the year? Or was this?
Yeah, this was February to June. Yeah. Yes, Laura.
I have one from Jill online. Do you think that the amount of clutter around your monitors may have affected your number of bat passes detected, possibly resulting in less passes in less urban areas, and possibly because more clutter in rural areas around your mics, thereby affecting recording quality?
Yeah, that's a really good question. Because yeah, sometimes the areas that the monitors were supposed to be placed in did have a lot more cover. I didn't see any really like concerning variability in the number of detections of each monitor. And I don't necessarily know if it would make that much of a difference, just because as we saw, like the canopy cover didn't really have any significant effect on the number of bat passes. So yeah, that's what I would say.
a bit more on the bonneted bat? I couldn't focus in on it on your graph.
Yeah, I didn't include the bonneted bat on this because we have the detections, but we don't know if they are bonneted bats or if they're cicadas, because they're the biggest one, and so their calls are in a frequency that the detectors can pick up as rodents or cicadas, whereas the other ones are higher frequency, so the monitors don't get confused.
So I don't know this literature well, but are more bat passes, does that literally mean more bats, or does it mean that the individual bats have to be active for longer? Like, can it be that the low urban areas, the bats get food really quickly and then just like hang out for the rest of the evening, you know?
Yeah, that's a really good question, because yes, the bat passes are one call. So it could be the same bat. It's not a proxy for abundance necessarily. Yeah, that's something important to note and to look at. I'm not. No, sorry.
It's usually used as a proxy for activity.
Yeah, so it's like their hunting activity. It's kind of predicted that if there's more activity in an area, there are more bats. But yeah, one bat pass is just one bat call. But it's common to use bat passes in that way. Yeah.
Can you distinguish bat species from your calls that you're recording?
Yes. Well, I can't necessarily, but the computer can.
Well, Nyctaceus humoralis might be interesting to study because my friend Fred Holmes, who was a lineman for Glades Electric, told me that every time he went up a pole and took off a bracket, bat flew out. So I said, bring me one. And he did. It was Nyctaceus humoralis. So you could think of a power line going through rural and urban, and there could be Nyctaceus on almost every pole. So that might be an interesting species
Yeah.
for further study.
They are pretty abundant. I know because they have them down in like the Everglades as well. They're found more like ubiquitously across Florida. Yes.
So are the species that you looked at known to vary in their movement patterns? So are some more directionally persistent, moving in single directions over a long period of time versus others, going back and forth,
Mhm.
recursive movements?
That's a good question. I know that the Molluskidae family in particular, they prefer to fly like really fast through open spaces, whereas the Vesper bat family, they're like very, very generalist. There's a lot of different Vesper bats, and that's not found as much, but the Molluskidae have wings that are like adapted specially for fast flight through open spaces. Yeah.
I have a couple of follow-ups from Jill, if somebody else, okay. How high were your monitors mounted off the ground?
They were 2 meters, or like, yeah, this is my head, and it was about here. I'm 6 feet tall for those online.
Okay. And Jill says, also, did you say you detected hoary bats? They've not been positively identified this far south?
Yes, we did detect dory bats,
Okay.
and those do not need to be manually vetted.
I'll give you some other notes and statements that other people had
Okay,
later. Yeah.
cool.